Bismillahirrahmanirrahim. Dear students, today we are going to study O-level biology chapter 4, chapter 3, that is about movement of substances in and out of the cell. And actually it is diffusion, osmosis and active transport. Uh, so first of all, diffusion. Do you know what is diffusion? It is the movement of substances from the region of higher concentration towards the region of lower concentration, that is along the concentration gradient, unless the concentration on both the sides become equal. So diffusion is the movement of any substance, maybe solid, liquid or gas, from the region of higher concentration towards the region of lower concentration, unless the concentration on both the sides become equal. So diffusion is the movement of particles from the region of their higher concentration towards the region of lower concentration. You may call down the concentration gradient, or for this purpose energy does not required, and this and from any surrounding energy due to kinetic energy of the molecules will be enough for the mixing of the molecules, so their own energy. Some substances move into and out of the cell through the cell membrane, and in some cases cell membrane may or may not be involved. For example, when we mix some salt, a sugar, or maybe ink in a water, beaker of water, so then it is diffused equally to the air, membrane is not required. But inside the cell, if material enter or leave, then it has to pass through the cell membrane. So diffusion is very important for gases and solutes exchange in living organisms. So diffusion is affected by the following factors like surface area, temperature, concentration gradient, and distance, etc. Now let's discuss examples of diffusion. Movement of oxygen from the lungs to the bloodstream, and of carbon dioxide from the bloodstream to the lungs in human. Number 2, the movement of oxygen and carbon dioxide into and out of the leaf. Number 3, movement of soluble food molecules from the ileum into the villi. Now, the factors which affect the rate of diffusion are thickness of the membrane. The thinner the cell membrane, the faster the diffusion. Basically, if the membrane is thick, then the distance will be more. So the movement will be less, and if the membrane's thickness is less, then the distance will be less, so the rate of diffusion will be faster. Next, concentration gradient. You may also call it diffusion gradient. The greater the difference in the concentration of substances on either side of the membrane, that is, greater the diffusion gradient, the faster the diffusion rate. For example, if on one side the concentration is 100% and on the zero on the other side, so then the difference between the two will be of 100. So the rate of diffusion will be very fast. But if the concentration on both the sides is equal, then the movement of the substances will not be there, or it will be equal on both the sides. Next, surface area. Increase in surface area to volume ratio increases the rate of diffusion. The surface area to volume ratio is its surface area relative to its volume. The larger the organism, the smaller is the surface area to volume ratio. Membranes are often folded to increase their surface area. Cells involved in rapid diffusion of absorption in the intestine often have their free surface membranes formed into numerous tiny projections called microvilli, villi, to increase the area of absorbing surface. The biconcave shape of the red blood cells provides a greater surface area volume ratio for rapid absorption of oxygen. Next, size of the molecules and ions. Larger molecules and ions diffuse slower than smaller ones. And next is the temperature. Temperature is directly proportional to kinetic energy. So, by increasing the temperature, the movement of the molecule is also increased and the rate of diffusion is increased too. Now, my dear students, we have to uh, discuss some of the experiments of diffusion. For example, if we take a beaker of water and put a crystal of copper sulfate in that, so this will uh, mixed into the beaker of water within few minutes. Uh, so the crystal of copper sulfate is 100% and in the surrounding water the concentration is less. So the copper sulfate will be diffused from higher towards lower concentration. Similarly, in the surrounding water is more, so it will move from higher to lower concentration towards the crystal of the copper sulfate. So within few minutes all the material will be homogenized. This will be because of the diffusion. Uh, Similarly, gaseous exchange. In the air, there is more amount of oxygen than our body, and our body has more carbon dioxide than in the surrounding air. So oxygen diffuses from the surrounding air towards the body, and carbon dioxide from the body moves out through the lungs into the air. Next, my dear students, we have to study about osmosis. Osmosis is the net movement of water molecules from a region of higher water potential or dilute solution to a region of lower water potential or concentrated solution through a selectively permeable membrane. Water diffuses through partially permeable membrane by osmosis, and water moves into and out of cells by osmosis through the cell membrane. Water acts as solvent in organisms needed for digestion, excretion, and transport. And osmosis on putting plant tissues and solutions of different concentrations will show different shapes. For example, plasmolized, turgid, and normal cells, etc. The plants are supported by pressure of water inside the cells pressing outward on the cell walls. Now, dear students, if we put animal and plant cells in different concentrations uh, of solutions, you can say hypotonic, hypertonic, and isotonic solution. You know, hypertonic solution is a concentrated solution, and hypotonic solution is a dilute solution, and isotonic solution is having the same concentration. So the plant cells will become turgid in the hypotonic solution, will remain same in the isotonic solution, and will be plasmolized, wilted within the hypertonic solution. But the animal cell show a small different behavior that in the hypotonic solution it will be bursted out. Firstly it will become turgid and then it will burst out because it does not have cell wall, whereas in the hypertonic solution the cell will be shrinked or keratinated. uh, okay, dear students. Okay. Hello, hello. Can you hear me? Yeah. Hello, अस्सलाम वालेकुम. Can you hear me? अभी तो म्यूट ही है. Hmm. Hello, hello. Can you hear me? Hello, dear students. Can you hear me? Hello. Can you hear me? Dear students, can you hear me? Okay. Okay. So, my dear students, okay.